Today we're working on an experiment where we're going to look at lettuce and see if we can illustrate osmosis in plant cells. So you can see I've got a couple different types of lettuce: scissors, salt, sugar. You could use other solutes like Epsom salts or flour. And I've got it all set up here with 6 different containers. You could do your own 3 containers. I picked out some basil from my garden. So the first thing you're going to do is put water into each of your containers. You want to use about the same amount. We used about half a cup of water here. So you're going to pour about half a cup of water into each container. We want to control that variable, right? So use the same amount in each cup. It helps if you have someone else to help you with what you're doing. So now that we've got half a cup of water in each, in one of the experiments we're going to see how salt water affects our plant cells. So you can see in the middle cups here I've got water and salt, and by salt we mean sodium chloride. So I've got about a tablespoon of salt into each of those middle cups, one for basil, one for the iceberg lettuce. Give it a good stir to help it dissolve. And then next we're going to add sugar to the next set of experiments. So again, about a tablespoon to be consistent with how much we're measuring out. A tablespoon of sugar into each of those 2 cups, so we can compare the effects of salt water on the lettuce and sugar water on the lettuce. Next up, we're going to stir that. Never move your spoon or stir anything between solutions. You don't want to contaminate it, but it's kitchen chemistry, so you can be just a bit more flexible here. So now we're ready for our leaves. I've chosen iceberg lettuce and basil. You want each of them to be roughly the same size, so either 4 leaves that are the same size or squares that are about an inch by inch of the lettuce work well. We're going to keep one in air as a control, and we're going to put 1 leaf into pure water for each of them. Make sure they get submerged. 1 leaf into water and salt. Again, make sure they submerge. And then finally 1 into the water and the salt, water and sugar, and make sure they get covered. So now they're going to sit here for 15 minutes. Get a friend to get a timer going for you, if you can. And I will give you some pictures of these as we go. So you want to watch for the color. You want to kind of feel your leaves as they go. Do they get limp? Do they seem to get firmer? Wilting is losing water and getting more floppy, right? So you're looking for any wilting that might take place in these 4 leaves also. So we're about 5 minutes in to these solutions. We're going to check in on them and kind of try to get a feel for it. You really, if you can, do this yourself, so you can do the touching and the feeling that I'm doing here. So those first 2 are about the same as they were. A little bit softer, just a little bit on the water and salt leaves, and water and sugar, not a whole lot of difference that I can feel. So we will let those sit. While we're waiting, we can get ready with some forks. Those are going to be used to fish stuff out when I'm done, and also a couple of paper towels to set the leaves on. 5 more minutes have passed, so we're going to check in on our leaves again. And again, touch can be kind of a cool way to do this, so that's kind of firm there. This one is able to be squished more, and same in the sugar water. So salt and sugar, the leaves have gotten softer. Still nice and springy with the iceberg in the water, and then more wilted, softer in the salt and sugar water. And again, instead of salt and sugar, you could use different solids. You could also use different leaves, like zucchini, succulents. You could try this out with whatever kind of leaves you can find. So now we've hit our 15 minutes. We're going to take our leaves out of the cups and see if we can tell a difference in their texture. Again, remember that if the leaf is soft, that means that water has moved out of it, which is called osmosis. when water moves from areas of low solute concentration to high solute concentration, that's osmosis. So we're seeing if we can see some of that at play with our different leaves here. And some plants might be more susceptible to it than others. So I've got a new fork for each solution so we aren't mixing those up. And again, this is a great one to do yourself so you can kind of actually feel it. I'm going to pick these up and show them to you so you can see the difference. So here's the ones in air. You can see the iceberg is a little bit flatter. The iceberg that was in water has absorbed water, it appears. It's nice and rigid. Basal has stayed about the same. The water salt seems to have wilted, both leaves, especially the iceberg, but they're soft, kind of squishy, not nearly as rigid as the ones that were just in pure water. Sugar also has wilted the iceberg and somewhat wilted the basal. So that wraps up our experiment. Take good observations, do it yourself hopefully, and thanks for watching.